Wednesday, and it's Wednesday afternoon, time for the data science seminar. Hi, the oceanography group as well. And welcome to our data science seminar from the University of Washington, eScience Institute. As part of the University of Washington, we acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot Nation. Today, we have another talk from our eScience Winter Incubator series. We have today with us Dr. Allie Chase. Allie earned her PhD at the University of Maine with a dissertation on methods to observe phytoplankton in the open ocean using optics, flow cytometry, and pigments. Allie joined the UW Applied Physics Lab in the summer of 2020 as a Washington Research Foundation postdoctoral fellow. During her postdoc studies, Allie is working on algorithm development to detect phytoplankton community composition from ocean color satellite measurements. So today, Allie is going to talk about her work that she developed with our data scientist, Valentina Staneva, here with us. And she will talk about user-friendly tools for oceanic plankton image analysis, also named UTOPIA. And please put your phones on silent and ask questions at the end, preferably. And Allie, please take it away.
Super. Thank you so much. Thanks everyone for being here in person or virtually. I'm really excited to share some of this ongoing work that I've been doing with Valentina at the eScience Institute with you all. So just a brief outline for today's talk. I'm going to share some of the introduction motivation. Why do we care about plankton imagery at all? Some previous work that was done that our work is really built on top of. And then go into some of the details of our approach for the data management, our workflow, and also our efforts to use cloud data storage and deep learning and cloud computing. And then of course, what comes next? As I said, this is ongoing work. So this is a satellite image, well, data derived from satellites showing chlorophyll concentration in across the entire globe. This is data from October 2021. And chlorophyll A is the major photosynthetic pigment found in phytoplankton. Phytoplankton are the microscopic photosynthesizers found everywhere in the world ocean, and they support our entire Earth system. They are the base of the marine food web. They produce oxygen as a byproduct of photosynthesis, which makes our atmosphere habitable for us and other respiring organisms. Phytoplankton also cycle nutrients and play an important role in the carbon cycle. And their distribution in space and in time across the globe is extremely heterogeneous. So you can see in this graphic that there are higher chlorophyll A regions in certain parts of the globe. And this is representing the total plankton biomass in the ocean. But all plankton are not equal. They're extremely diverse taxonomically and functionally. And the different types of phytoplankton play different roles in cycling nutrients, supporting higher trophic levels in food webs. And maybe you've heard of harmful algal blooms and red tides. So there are many reasons in ocean research that we want to know where different phytoplankton communities are found and how their distributions may be changing. There are models that are used to represent this kind of information that we're after. So this is one model that comes out of a group at MIT. And the different colors are meant to represent here not total plankton biomass, but the relative presence of different types of plankton. It's a beautiful graphic and it's highly accurate in many of its physical oceanographic modeling capabilities. But in terms of the phytoplankton community composition it displays, it's based on real data, but we also know that it's not totally accurate in many ways. You can see there are large bands of the ocean that are sort of assigned to one group. And we know from our in situ observations that the biology is much more complex than that. So improving these models or using them to address certain questions is really limited by how well we know what's actually in the water. Just to bring it a little closer to home, here's a true color satellite image of the Salish Sea waters. And you can see there's some variable shades of, I hope you can see, yes, good, of blue and green and different brightnesses. And this is due in part to the presence of different types of phytoplankton that are found in the water. So how can we assess what's actually in the water in terms of types of plankton? Traditionally, we collect water samples and then maybe look under the microscope, and a taxonomist would count the presence of different types of plankton. This type of analysis is still extremely valuable in many settings and for answering different questions. It's also very time consuming, and the ocean is enormous, as you know, so it's ultimately limiting in how rapidly we can collect data. But technology is advancing. So these images are plankton that were collected with an imaging FlowCytometer. So specifically an instrument called the Imaging FlowCytobot, or IFCB for short. Was developed about 15 years ago at Woods Hole Oceanographic Institution on the East Coast, and since been commercialized. And it's a FlowCytometer, which means that it observes particles one at a time. Traditional or conventional flow cytometry enumerates particles. This is an imaging flow cytometer, which means it also takes a picture of every particle passing through the flow. We deploy it on ships. It can be deployed on moorings. For my research, I use data where the IFCB has been deployed on research vessels. And it's picture, no, it's coming later, sorry. It's set up in the lab and it's attached to a flowing seawater system. And it's designed to draw a sample of about 5 milliliters a few times an hour, and then pass the water through the fluidics and capture a picture of every plankton found in the sample. And in this way, it can record thousands of images in an hour or millions over the course of a multi-week expedition. So a lot of data has been collected with different IFCBs. In particular, I'll be referring to data that was collected by an IFCB belonging to the group where I did my PhD at the University of Maine. These data I'm showing here are displayed on a dashboard that's a web interface designed also at Woods Hole, where they host several different IFCB data sets for visualization. These data are from the Western North Atlantic, part of a multi-year mission, and these data in particular are the ones that I'll be referring to in the work that Valentina and I have done so far. But in addition to the North Atlantic, where we collect about 5 million images, there's also data from the North Pacific, so off the coast of Washington out into the North Pacific. And then just last summer, some data collected here in the North Atlantic, Eastern North Atlantic. So these are several different data sets that have been analyzed to different extents so far, and the data continue to be collected. So for example, I just returned about 2 weeks ago from a 2-month expedition crossing the South Atlantic Ocean with the same IFCB, but this time on a research sailing vessel called the Tara. We traveled across a wide range of water masses. You can see the background color is the water temperature, ranging from just a couple degrees Celsius up to almost 20 by the South African coast. And I thought I'd add this picture, which is this very small lab that I worked in on this sailing research vessel, and the arrow is highlighting the IFCB. It's about 1 meter tall. So it's connected to power, and then it's a bit hard to see in the picture, but just above the IFCB, there's a tube, sort of a whitish opaque tube, and that's the flowing seawater that's pumped continuously from the surface waters past the IFCB, where it draws the sample from there. I should also mention this is an amazing instrument that was originally deployed on moorings, and it's designed to be, requires power, but it's designed to be left for months at a time on its own. So it's fully self-contained with the way it cleans and calibrates itself. So it's not cheap, but it's really an amazing piece of equipment for oceanography. Here are some screenshots that I grabbed during this recent trip, just to show you examples of how morphologically and taxonomically diverse the plankton are. So these are all to the same size scale. You can just see there's a variety of sizes and also different populations. For example, on the left panel, there was a fairly homogeneous community of one type of diatom. And we had some small cells in more kind of the middle of the ocean, and then some larger cells closer to the coast. So I hope I've convinced you at this point that we need some tools to deal with these data. They're massive data sets, and the questions we could answer with this type of information collected at high spatial and temporal resolution, and across dynamic ocean environments are numerous, but there's, in my opinion, there's a bottleneck at this point in our ability to really assess the data in an agile way, where we can sort of pick up a data set and use it without having to spend too much time trying to figure out what the cells are. So that motivated my participation in the data science incubator program, which has been really positive experience. So I recommend applying if anyone's thinks they have a data set that needs they need to work on. So our two main objectives, which are both ongoing, but we've started to tackle both, certainly, are to train and validate a machine learning neural network using IFCB data, and to share this network in a public open source way and with clear documentation. So there has been work done using different machine learning different machine learning networks and some deep learning for IFCB imagery, but they're generally have been conducted in situations where it's specific to a certain data set. That can be useful, but I think the time is right for tools that are more open source and collaborative, as is generally the trend in earth science. And I think it's really powerful and exciting. So first, I also just want to briefly share what's been done before, because I think, it's important to appreciate how much effort went into these data before I even started this incubator project. And just to share how we actually classified them, because I did start the data incubator project with classified plankton imagery, and that's how we trained our deep learning networks. So previously, we took raw data from the IFCB, we processed it to define or extract image features. So that includes finding the outline of the cell within a picture and area estimated or volume estimated of that image. The data were then uploaded to EcoTaxa, which is a web-based software to visualize plankton imagery. I'll show you what that looks like in just a second. And a learning set is manually built, where a researcher will categorize what the plankton are, maybe 200 of each type, so that the software has somewhere to start. And then a random forest machine learning algorithm is applied using that learning set to classify all the images. It's a good start. It's better than nothing, but none of the output or the outcome of that random forest machine learning is good enough for use in scientific analysis. So just ballpark, on average, it would be about 50% accurate in classifying our plankton images. So it was a start. But then we would check manually all of the images that were classified by the machine learning network and either confirm or correct their assigned category. Then export the information off of EcoTaxa. So a unique ID for each image based on date, time, and an image ID, and what it was categorically assigned as. And then we can use the data for either for deep learning or for various analyses. This is just a screenshot of what the EcoTaxa platform looks like. So it's a really nice visualization tool, but it is sort of a click and drag to categorize plankton cells. Over the course of 2 years, myself and sort of a team of students, undergrads that we employed, validated to about 2 million images. It's a lot of data, but there were 5 million just from this expedition alone, and that took us years. So we need deep learning tools that can do better. We'll always need some humans in the loop, but a deep learning network that can assign categories to plankton images with relatively high accuracy will be extremely powerful. Just to give you an idea of some of the work we did previously to define the different plankton types. So here are some beautiful images of all different categories. In the end, for our deep learning, we have used previously and currently using sort of 7 to 10 taxonomic categories. But that's an avenue of exploration, is how many categories to use. I worked with a colleague a couple of years ago, and he built a deep learning network to assess these data from the North Atlantic. We met with him at the start of our work, and it's been hugely helpful to build off what he did. But as I mentioned previously, that was a project that was sort of self-contained for that data set. And our goal is to build and provide tools that allow application of neural networks sort of without reinventing the wheel each time. This is an example of a confusion matrix, just to show these are sort of the main plankton categories, plus a couple of non-living categories, but that we use. So the predicted category across the top x-axis and the true category. So what you want to see is a high value across along the one-to-one line there. The confusion matrix is also useful for seeing where the networks have made mistakes, but we'll come back to this with some of our data as well. And then the last slide for the previous work, just to show that when I plotted all of the network results from these two oceans, you can see there are major taxonomic differences. So there's sort of yet unexplored questions about phytoplankton communities on major ocean scales. And I'll just say that I applied the previously trained network to this North Pacific Ocean data, but it does need some further analysis or application of the network we're working on now. So our approach, we met with Emmett, as I said, to build off what he did. And then we thought a lot about how to set up our workspaces. We used Google Drive, also we use GitHub, and then the Microsoft Azure portal, which I'll come back to. I spent some time exploring Python machine learning resources. I'm a MATLAB user, so I am making the transition to Python, or at least adding Python. And a shout out to the software carpentry at UW group, which I had an excellent experience participating in a software carpentry workshop in January. Highly recommend, through the eScience Institute. We downloaded the data and we're exploring and explored some cloud options, as I mentioned. And then the first sort of, real hands-on step that we did was to set up a test case where we could develop a workflow, but just using a small subset of data. And this seemed fairly straightforward. We have PNGs, so those are the plankton images. And then we have a CSV, so a big text file, and every row represents an image. So we have a unique identifier for each image. And then we have the category, so diatom, dinoflagellate, and that's, as I just shared with you from the previous work that had been done. So we just need to pair these two data sets, and then we can pass them to a deep learning network. We were working for this part in Google Collab, so Collaboratory, which is a Google environment where you can work in Python and write and run Jupyter notebooks in like a shared drive space. So that was helpful for when Valentina and I were working asynchronously. But even at this first step, we had some challenges. And I'm going to try to share the things that hung us up throughout this. I hope, in the hopes that maybe there'll be something that's a useful takeaway for other big data users. What we didn't know is that you can't upload files of many small images to Google Drive. They sort of limit it. So we couldn't click and drag our folders there, even though the data are not actually particularly huge, especially a subset that we're playing with. Valentina found a solution using rclone, and then we were at least able to get a couple of days of data onto the Google Drive. So we started using data from one day. This is right near the beginning of the first research cruise in the North Atlantic. There were 66,000 images on this day. We ended up subsetting down to 10,000. And so here's sort of our initial data workflow in the Google Drive collab space using Python and Jupyter notebooks. So we had, again, our CSV file. So this is one row per image. We had the CSV file representing all North Atlantic labeled data, which is about 2 million images. We added a column to this file representing the PNG path because we know where the data are located in our drive. And then we built a simpler data frame just to show the category of the particle, the PNG path, and sort of the local path. And then we added a third column showing the entire path. So just to simplify, and I'll show you an example of what that looked like. And as I said, we subset it down to 10,000 images. So here's our very simplified 3-column data. So we have the PNG path. This is just defined by a date time stamp of the folder where from the IFCB sample and the image ID. The category, in this case, these 5 first rows, 4 of them were identified as other, perhaps a label that could be improved. It's really unidentifiable cells and 1 dinoflagellate. And then the full path of the PNG. These are 10,000 test images. You'll notice that the distribution is highly not equal across these groups. In particular, in this case, there are many other unidentifiable cells. These are data from November in the North Atlantic when there are, well, in this case, in this sample, there were many small cells which are harder to identify in the IFCB imagery. It's not totally atypical to have a majority of unidentifiable cells in IFCB data, but this is a more extreme case where there are really a lot. On the one hand, we talked about curating sort of a perfect data set to play with. On the other hand, it's kind of nice to use realistic data that have this issue of uneven distribution because that's what the real data look like. So it's something to consider. We also will in the future be playing around with some upsampling and downsampling so that we can even out the distribution. So then another major step in using the IFCB data is image preprocessing. In this case, the networks we're using require square images. So we had to resize and then pad the images. There could be possible future avenues where we don't have to square the images. There also may be some implications of some of the resizing where in these examples, it doesn't stand out, but for example, a long skinny chain or phytoplankton cell could be sort of squished when you resize it. And so that change in the ratio of length to width maybe has an effect on the network performance. So after we pre-processed all our data, then we ran just a very simple Keras deep learning network. This was based on just basically copying and pasting like a deep learning model for digit identification. So very simple, basic. And the nice thing about the way it runs is that you can see as it's going each epoch, which is each iteration of the deep of the neural network, you can see the accuracy changing visually. In this case, we had loss in accuracy at the end of 0.6 and 0.8. The goal is for the accuracy to be as close to 1 as possible. And this, in this case, is total accuracy of the entire network. There are other ways to explore the accuracy of each group. And then also what can be important, as I alluded to earlier, is that when something is mislabeled by the network, what was it labeled as? And that's information that can be really valuable to us because, for example, if the network is wrong half the time on one category of plankton, but it's always putting them in one other location, that's information that we could use maybe in how we prepare the data or design our network or use the data at the end. So at this point, we needed to transition to cloud data storage. The Google Drive would not be sufficient at some point for all the data. We are using Microsoft Azure blob storage. The nice thing about this is that a few things. It's accessible from different computing environments, but also it's easy to change the code, for example, in a Jupyter notebook to point towards the data on your local Google Drive or on the cloud. So there was only one line of code where we had to put in an address basically for the blob cloud storage and point to it. And we used an access key for that, but also there are different options for your data being public or private that may be relevant for different applications. One thing to note is that in this case, this blob storage, I guess they call it blob storage in part because it's not a typical file folder hierarchy like we think about normally. So the data sort of all together, and we were presented with a challenge where we couldn't query and search within our data the way we were used to being able to do. But there is, the solution we found to that was to use our CSV, so our big inventory table of all the samples. So there were challenges that came up with setting up this environment. I would say that there's some room for improvement in this portal. But in the end, it was doable and it's working. And we also then transitioned our computing environment to Microsoft Planetary Computer, which, and I circled here the hub, which is what we used. Microsoft Planetary Computer is a platform where you can log in and use computing resources for free from Microsoft. It talked easily to the Microsoft Azure cloud storage, which is one reason we used it. Also, there was some access to free GPUs. Hence the little GPU sign there. And we could easily connect with this connection string in the notebook. So the connection string was provided in the Azure portal. We put it in our code in the Planetary Computer, and then it was pointing to the data. I'll note there's no local storage on the Planetary Computer, so that comes into play in how we managed our data. And I'll come back to my next point later, actually. So this is our workspace setup on the Microsoft Planetary Computer. You log in, you make an account, a Microsoft account, you log in, and then you're ready to go. So they have many pre-installed, we were importing some Python libraries, but their environment is sort of ready to go for a lot of research related to earth science, but more broadly could be useful as well. There's a place you can open a terminal, you can use Jupyter notebooks, and then we also have some Python modules for our different networks and utilities, so data, different functions that we have been working on. So as I mentioned, there's no local storage on the hub, but that's okay because we were transitioning to do our deep learning data preparation in an on-the-fly style. So this is a scalable machine learning approach where rather than reading in all the PNGs and building a giant array, say, of all your data, when the network is defined, it's pointing to the data. And then as the images are read in for the batches of the neural network, the deep learning network as it runs, that's when they're pre-processed. So this refers to the reading in and the preparation of each image to be ready for the deep learning network. This is scalable and useful because it doesn't matter how many images you have, you're not going to be limited by RAM. It's sort of accessing them on the fly. It can be slower at each read compared to reading in everything at once, pre-processing, and then you have your array ready to go. But on the other hand, it works really well for large data sets. So just something to think about when you're handling data such as these millions of images. Do you want to have them all in memory, processed and ready to go, or sort of point at them with, in our case, that's what I refer to when I say instantiate an image generator. So we sort of defined within the code, the pre-processing functions and the neural network functions that would be pointing at the data. We used an 80-20 split, so 80% of our labeled data for the training and 20% for the validation. And then we define the model. So I wanted to show this for people maybe haven't done deep learning. I had only dabbled a little bit before I started this this project for the data incubator. And of course, it can be sort of infinitely complex getting into deep learning, but to start, it can be also very simple. So we used Keras, which is a library of software of functions used for deep learning and open source. And this on the left, where you see the defined function create CNN model, A1. This is actually from Emmett. We made some small changes, but it's built off of one of his model functions. This is the whole model. So these are the layers. They're convolutional layers sort of in the middle and the inset there, and it's looping through and doing different convolutional mathematics there. And then down below is where you kind of have the layers where the data are sort of assigned back to being real values. And then on the right is the Keras model function, and this is where the model's actually being trained. So that's all the code for training, for defining and training the model, not the data preparation, but and the train, gen, and validation you see in the blue box on the right for the model function, those are referring to where the image generator was instantiated. So those were defined previously in the code, and that's sort of where we point to the data, and we define the model, and then we fit it, so we train it. This was working well. We were able to run our same 10,000 image test case using the Planetary hub and the pointing to the blob storage for a time, and then we started having some problems with the Planetary hub in terms of being able to log in and getting kicked off. And so the free GPUs are great because they're free, but we transitioned more recently to using Azure virtual machines. So this requires cloud computing credits generously provided by eScience. This requires setting up your own virtual machine, and Valentina has been doing a lot of work on that, but they have a lot of built-in things sort of ready to go. So if you're thinking about cloud computing, there are a lot of, there's a lot that's sort of already set up on the Microsoft Azure virtual machine space. So it's relatively painless to get it going. And the codes that we had sort of ran. So they have a lot of things that are pre-installed, ready to go. So then we could run our same Jupyter notebook now, but with using the virtual machine. So the cloud, using our cloud computing credits, we could use this virtual machine and sort of more robustly run our neural networks. Then we use Tensorboard to view the results. So this is, will this work? Maybe not. Maybe I can just do this. This is a powerful, oh no, it wants me to stop sharing. Okay, I can just do that, sorry. This is a powerful way that you can visualize different experiments with neural networks. See if I can find my computer. Okay. I'm coming, guys. So this is Tensorboard. So this is a Google product, but it's free open source software, and you can relatively easily connect to it using the output from your deep learning network. I'll just show you one recent example. So on the x-axis, hope everyone can see, are the epochs, accuracy, and loss, so training and validation. And so it's just a nice way to visualize, as you can see, as your network is asymptoting, because well, it's reached the point where it can do the best that it will. And also what's really great is that if you're going to test many things, you can see there are a lot of different versions here where we were experimenting, you can toggle them off and on and have a nice visual, because you can imagine if you're going to experiment and run your deep learning network many different ways for a variety of reasons, either with the way you've prepared the data or the way you've designed the network, or both. You want to be able to sort of keep all of that organized. And in this case, they're labeled by date, so you can keep track of your experimentation. Also on Tensorboard, there's this tab for images. So here are example images, and they've been labeled with the true and predicted. So you can kind of have a quick visual and scroll through and learn something from that. So let's just go back to the top. Thanks for your patience. So if you haven't heard of Tensorboard, I recommend it. All right, are you with me now? Sorry for that screen. So just a little bit thinking about what's coming next. There are some different data science ideas for the model runs. So there is this option to kind of have the simple off-the-shelf neural network approach, but then there are also a lot of ways that we can iterate on that and hopefully improve our results. Data augmentation, rotations, and replicates. So this refers to how we prepare the data. For example, I talked about the upscaling or upsampling, sorry, when we have only a few of certain types of images. Also, this issue of preparing the length and width ratio when the data are prepared and we're squishing them down to squares. And tests to evaluate the impacts of doing this to the images. Also background color and normalization. So the data that we have is all currently that we're using is all from one IFCB, although even within one instrument, there can be differences across expeditions depending on the settings of the instrument. But there are quite a few researchers who collect IFCB data or even open ocean continuous IFCB data. And so I just added a few images to this slide of some other researchers with IFCBs. And so you can kind of see the difference in the background color, some different shades of gray or even different across the image. And maybe playing around with that, the background color is a different version of augmentation, because especially thinking about making a tool that's useful for someone to just pick up and use with their new data set, you want to be sure that that kind of issue wouldn't completely be sort of a deal breaker. So we're trying to think of all these things that can make it more broadly applicable. Also model experimentation. So there's the data preparation side, distribution of categories, etc. But then different ways that we can test our models, how we define plankton groups. Maybe we need to consolidate some groups. We could try separating and running across more groups. There is this major issue of the unidentifiable images. So that's an entire project on its own and has been in the past, as I'll come to in a second. Also applying a trained model to completely independent data sets. So, so far we've been sort of using the same labeled data, and it's from this one expedition to train, validate, and test, but it will be obviously important to test on completely independent data sets. Playing around with some of the network layers, like I said, we're currently using relatively simple network, although I will note that kind of in talking with Valentina and learning more about our challenges, I think that in many ways the sort of messiness of the data itself is a challenge as much or more than tweaking the network. So maybe the network layers need to be changed and that could help, but it's not magic. So if the data are poorly labeled to start or just poorly distributed or whatnot, then you know that will limit how well your network can do ultimately. So kind of trying to think about both things, the coding network side, but also the data side. A two-stream model, as I referencing here, is something that Emmett tried, where he first identified to separate, he first trained a model to separate the unidentifiable from the identifiable images, or living and non-living, and then a second tier of the model where he was then just categorizing the living and identifiable, sort of the bigger plankton usually. So there are different ways that we could approach that. And then unsupervised learning of unidentifiable images. There was a summer project several years ago from a student in France, and he took all of our other images and did some unsupervised clustering. And these are 3 of the clusters that came out of his work. You can see all the cells within each cluster are fairly uniform, but they were all put in the unidentifiable category. So there may be ways we can use that kind of unsupervised learning to further sort through our cells. So this is our GitHub organization. This is in the vein of our platform for open source software development collaboration. It's in the works. So this was where there'll be all the documentation and sort of instructions, also the caveats and disclaimers. You know, it's open source and it's GitHub, so we know that people can do what they want with it. But I'm also hopeful that through like clear documentation, we can provide networks and information for people as a place to start. Of course, taking someone else's trained network off the shelf and applying it to a different data set, you'll never know how well it really did on your data set unless you take the time to look manually at some of the images. So there are considerations like that. This won't be a network that will just be provided and will be usable perfectly for all future data sets, but it will be a place to start, and hopefully also just a forum and a place for people to collaborate and share ideas. So some takeaways and next steps. The workflow and the data setup and the cloud computing storage and setup takes time. I just make this point to say that I don't think it surprised Valentina. I think it took more time than I thought, especially with how much work I had already done with these data and others had. But you know, the data manipulation, as I assume many or all of you know, there are always bugs and issues and problems, and real data from the environment is messy. So Valentina and I try to embrace the idea that that part of it is fun too. And yeah, so we try to have fun with all the aspects of data science, even sort of the traditionally tedious work, because that's really where I think you can make or break the outcome of your results if you're thoughtful and careful with how you set things up. 2 major avenues going forward for our UTOPIA GitHub organization. Sort of just touched on this, but code and documentation for people who want to apply an already trained deep learning model. Maybe they don't have the resources to train their own. So predicting on your data takes much less computing power than training a new model. And also people who may want to train their own models, just some resources to see how we did that. As I mentioned, a public space where people can collaborate, and systematically testing different network structures and data pre-processing variations. And hopefully within, I don't know, a month or 2, we'll be able to have a network that we can try applying to all of the data that I just collected last month in the South Atlantic. It's untouched so far, so it's a huge raw data set, but if we can try running the deep learning network on it first, then we can push it to ecotaxa to visualize and maybe correct some errors, but it will be hopefully a great first pass at the data so that we'll get a huge jump start on doing ocean science with it. There are also a lot of opportunities for student participation in our group and with this project. So if you know anyone interested, happy to collaborate and get more people involved in working on this stuff. So thank you so much for your time, and the Washington Research Foundation for funding my postdoc, also for support from eScience Institute, my collaborators, and a huge thanks to Valentina, because it's just been such a pleasure to work with her and continues to be. So thank you. Happy to take questions. Yes.
Is it possible to encode, like, I know you're using a CNN, but is it possible to use like other environments for other data along with the model, such as like the data?
Yeah, great question. Okay, so the question is it possible to use other parameters along with just the PNG, just the image itself in training a model? Definitely possible, something that we have tried. So Emmett included in his model, he tried a system where he had all the vector data. So he was actually using the extracted image features like size, area, biovolume, and then we included lat, long, temperature, and salinity. And there is chlorophyll data actually because it, as you know, the images are fluorescing and that triggers the camera, and that data is recorded. So yes, there are ways to combine both just sort of the image side of the deep learning of the CNN with vector data. One hesitation, I guess, or one reason, I mean, because why not? Why would you not just throw more data? One reason we thought of that you maybe would want also a version that just takes images is for a future user or someone else that just has images. Maybe they don't want to, or for whatever reason, aren't going to run lots of image feature extraction code on their data. So like we're sort of starting with the simplest, like here are just the images. What does it think they are? And then definitely like sort of a parallel version where you add other features would be awesome, because I think it probably could do better. Yeah. Yeah.
Did you start out with an accuracy if you wanted to reach, and did you have like a goal in mind? Because I'm thinking like that's about as good as even biologists do when they're looking at a microscope to get the agreement between multiple of them. And then similarly, did you discuss like false positive implementation versus false negatives? How do you trade off between those?
Yeah, great. So the question is, is there an accuracy we're trying to reach, and how do we sort of deal with false positives and false negatives and those considerations for the accuracy? So in terms of an accuracy we want to reach, it's a bit arbitrary in the community, I'd say. In other talks, I've seen people discussing this kind of thing. It's either sort of an informally agreed upon accuracy of like 85% accurate. Of course, depending on your question, it matters. So what are you trying to do with the data at the end? Yeah, maybe I shouldn't even throw numbers out there because, you know, are you trying to calculate the exact quantity of a harmful species, or are you trying to ballpark estimate the carbon found in one major group? So it does depend on your question. But side note, your point about it being maybe, you know, as well as humans could do, we actually found that the network that Emmett had trained corrected quite a lot of human error in the data. So the results of his network, the wrong answers, he took a random selection of them, and then I went through them all again carefully. As I said, we had many students working for us and probably some tired hours that I spent doing it also. And many of the errors in his model were actually true, but we had labeled them incorrectly in the first pass. So that's kind of a fun and interesting piece of this, is that there could be a situation where the network is correcting human errors. And yeah, we do definitely consider like the false positives, false negatives, because that also depends on your question, like, and what you're trying to get at. But there could be a situation where, you know, the second most likely option or classification is also really valuable information, because, you know, or how quickly does the accuracy drop off? Maybe it's, well, it's 60% likely to be this, and then, you know, 30% likely this, and then drops way off. Or maybe it's, you know, so the distribution of how accurately it's categorizing images and what it's miscategorizing images as is, yeah, plenty of exploration to do there. Thanks. Should I be looking on Zoom, or people?
There are no more questions.
Okay.
Thank you, Ali, again, for your participation. I'm actually sure you had a nice experience with our program.
Yes, very positive.
Thank you.
Thanks everyone for coming.
Thank you. We'll see you next time.
Come see me if you want to work on this.
Yeah.
Looking at you, oceanographers. No, just kidding. All data scientists.